And we are ready to introduce the second speaker of the session. This is Gerhard Hummer from Max Planck Institute of Biophysics and Frankfurt University. Gerhard will give us a talk entitled Molecular Dynamics Simulation of Ion Transport. So it's over to you, Gerhard.
Well, thank you. Thanks for the introduction, and thanks for the invitation to present. Can you see my screen? Well, I assume so.
Yes, we do.
Yeah, fantastic. Great. So thanks for organizing this great meeting and yeah, putting up with all the conditions and restraints that we're dealing with. I think this is a very nice way for us to share some of the science. And so I will be talking about molecular dynamics simulations of membrane transport. So this will have a focus on active transport machineries and not ion channels. And I hope you will find it interesting, and if not, at least bear with me. So motivation, I don't think much has to be said to this expert community in this meeting. Obviously, lipid membranes are serving many important biological functions, from being barriers to surroundings and prevent infection and entry into the cell, to dividing up the space of the cell into compartments, and even some of the more interesting aspects of using the effectively capacitance and division to store energies in bioenergetics. As a result of these diverse and important roles, elaborate transport mechanisms have been evolved to deal with critical cellular functions. And these transport mechanisms involve ions, metabolites, but also you need to transfer information in the form of signals. So in my presentation today, I will be looking at molecular dynamics simulations as a tool to gain an understanding of the function of some of these transport mechanisms. And I will start out with just giving you a glimpse at an exporter in action using quite standard default MD simulations, albeit with quite a bit of effort in setting up the unique chemistry of an RKL system. I will then, in the main part of my talk, use a simulation system that is designed for rare event sampling to look at the exchange of ions and protons in a sodium proton antiporter. And in the third part, I will look at this system in a bit more detail from a thermodynamic point of view to try to understand the substrate specificity in more detail. And I will conclude more with an outlook of emerging challenges in this field and concentrate on some recent work on ATP-driven transport in an ABC exporter. So just as a reminder, the transporter through membranes, active transport through membranes is usually associated with the alternating axis, a model that has a long history. And here in this cartoon, taken from a paper by Beckstein and Drew and collaborators, you can see schematically two of the putative alternating axis mechanisms, where you have on the top, you have a rocker switch type axis where two domains toggle about a common the center, and in that way change axis. And in the bottom, you have another model where you have one of the domains move up and down in an elevator-like fashion, and that way carry the substrate from one side of the membrane to the other by sliding it past a sealed gate at the center. So with this in mind, we can have a first look at the transport process, and this is work done in collaboration with the groups of Shara Safarian and Hartmut Michel here at the MPI of Biophysics. Their groups have solved the structure of a multi-drug resistance mate family transporter, and they have now structures in both an inward-facing and an outward-facing conformation, and these are shown here at the backbone level with the membrane schematically indicated by the dashed lines. And you can already see that these structures indeed have the access to different sides of the membrane quite nicely visible. So by using standard method to give at least a rough idea of a large-scale conformational change, and this is targeted molecular dynamics, TMD, we use these two structures as beginning and end points to look at a possible pathway for how this conformational change from an, in the axis state could happen. And here you see an MV trajectory. I hope you can all see the movie of this transition. And in essence, it is despite the overall very dramatic change at the local level, changes are actually quite modest. It's the formation of new contacts and the breaking of old contacts, and a transition through an effectively occluded or sealed structure that ensures an intact membrane for the overall transport. But how is then the substrate taken along for the ride and released taken from one side of the membrane and released on the other side? And here again, MD simulations gave us a glimpse of what is happening. And what we saw in the MD simulations, these were carried out by Reza Mehdipour, really an outstanding postdoctoral fellow who is actually now in the process of starting his independent career. So what Reza found was that spontaneously, one of the lipids from the membrane domain diffused into the veg of the transporter and flipped its orientation and became trapped in this upside down state. And turns out that this is backed up, this lipid binding and flipping activity is backed up by the experiments where they have seen evidence for at least strong lipid interactions. Just as an aside, I want to note that this is an archaeal transporter in a hyperthermophilic archaea. And so the simulations were actually done in archaeal lipids with isoprenoid tails. So this hopefully shows to you that with relatively standard MD simulations, we can get already a good understanding of how transporters function. But now I would like to go a bit further and look at the sodium proton antiporter in a bit more detail. And antiporters are used to control cellular pH, but also salt concentration, and in particular volume, because sodium acts as an osmolyte, whereas protons tend to be buffered. In that way, one can establish a volume control. And so we have now the structures of a number of sodium proton antiporters, and recently we also obtained an antiporter, or actually several antiporter structures of the so-called CPA1 type family, which are non-electrogenic, where you have the exchange of one proton against one sodium ion. And so we have structures of one of these, both in an inward-facing and in an outward-facing conformation, albeit at only low resolution in the outward-facing structure, which comes from from electron microscopy map. And so if one goes now back to this picture of sodium proton antiporters as machines with alternating axis, here you can see we're going back to the cartoon of Bechstein and Drew, and back it up with one of their structures of the NAP-A CPA2 type transporter. And there you can already see they have very strong and very convincing evidence for an elevator type motion, where this domain here on the right is moving up and down by several angstrom. In our structure, this is the structure of APA. What we have is a much more gentle motion in this transporter domain. We see about 3.5 angstrom movement of this helical bundle up and down, associated with about 10 degree tilt in the domain. So motion is on a much smaller scale, appears much gentler, and this is illustrated here by showing the vertical displacement on the vertical axis and the rotation angle on the x-axis in simulations in the inward open and the outward open configurations, and you can see about 10 degree rotation, about 3 angstrom vertical displacement. Nevertheless, the simulations confirm that the two models actually have an alternating axis. We see spontaneous ion binding from the inside and from the outside into the respective conformations to the residues that have been identified, also based on a thallium-bound structure of this transporter. And so this confirms that we have good structural models, but and sets us up for MD simulations of the actual transport event. But the problem is that we know from the experimental measurements that the transport rate is in the minutes regime, is seconds to minutes regime, with about 4 ions transported per protomer per minute. So this is obviously a very slow process, far outside the reach of MD simulation. So we could use biasing schemes, such as pulling domains up and down. But the problem, of course, is that any kind of strong bias, in effect, presumes the mechanism that is used by this machine to exchange the ions. So what we did instead is we tried to adopt a simulation technique for rare event sampling, where one is not biased by exerting external forces on the system, but instead one makes judicious choices of initial conditions and lets them evolve in a completely unbiased fashion on the original free energy surface, and then tries to sample rare events through a clever trick called transition path sampling. So the sampling mechanism does not require a priori knowledge of the relevant reaction coordinates, and transition events, in this case transport, are sampled exactly as they would be in a very long equilibrium trajectory. So this is a very powerful, very promising technique, but of course, as with every technique, it has also its downsides, and that, as I indicated, is finding states where if you propagate them forward in time, you can actually see transition events. So to give you a bit of the background, of the theoretical background also on this transition path sampling, there's transition path theory. And I know we have Eric van den Einden here in the audience, and he's one of the experts in this. And here you see the fundamental relation of transition path theory that brings together the Boltzmann distribution, the equilibrium distribution on the left, and the distribution in the transition path ensemble, that is restricted ensemble, looking at only transition events, which are connected to each other by, on the left, the product of the so-called committers. The committer is the probability when you initiate the trajectory at this point of reaching the forward state in phi A, or the backward state, the starting state phi B first. And also on the left in the denominator is the time, or the mean time of mean duration of transition path. And on the right, you have the lifetimes in the denominator in the respective state, in the beginning state and the end state. These are the rates, or more precisely, these are the fluxes between the two states. So we have in one nice compact relation equilibrium, rates, committers as a form of reaction coordinates, and transition path times connected. So how do we make use of this? So what we need is, in this transition path sampling, is we need seed path. We need a good starting point from which we can initiate the sampling in this trajectory, or in this path space. And the seed paths that we generated were coming from targeted MD. We used two seed paths, one targeting forward by going from the inward to the outward open state, and one backwards from the outward to the inward open state, keeping in mind that in this way we will have opposite hysteretic effects. And I will show that in a bit more detail later. And then we simply shoot off unbiased trajectory pairs from points along these biased paths, and do this in multiple places until some point we cross over from one side to the other, and in this transition region we can, with a bit of luck, find one point that connects to both states. And this is indeed was indeed possible. And then these initial trajectories are then used as seed paths for path sampling. So one way of thinking about this is as shooting from the top, where the shooting points, or the points where trajectories are initiated, we think of them as being initiated near the saddle point, near the transition state of the reaction. And indeed, there is a formal way one can use this intuition to make shooting from the top quantitative, in the sense of producing transition paths with the same statistics as one would obtain in very long equilibrium trajectories. And so we, in this way, we can get a very high success rate if we actually know, to some degree, the relevant coordinates. So how does this work in the context of the transporter? I already said we have a vertical displacement of this helical bundle, and we have a slight rotation. So the shooting range, indicated here in magenta in the bottom right, is expected to be roughly a line separating these two sets of dots, corresponding to inward and outward open conformation. So it's an intermediate regime in angle and vertical displacement. As I already said, we have a problem. We will be able to collect only relatively few transition paths because, as you can expect, these is a rather large, complex system. This is a few 100,000 atoms. The paths themselves take many tens of nanoseconds. So it will be challenging to collect more than a handful of transition paths. So how do we ensure some degree of convergence? And as I already pointed out, by taking advantage of the fact that the initial path created by forward TMD and by reverse TMD will have opposite hysteretic effects, we can look at whether the transition states harvested from these two paths are coincident, which would indicate that we have indeed found a reasonable mechanism. So this is what we did, and here are states where we actually were able to shoot off trajectories that reached the forward outward open and the reverse, the inward open conformation spontaneously without any external bias. And so in this plot on the right, you see location of these transition states in between the two islands of gray dots corresponding to inward open and outward open conformations. And you see, in addition, connected by arrows, the conformations of these transition state structures with side chains and water molecules illustrated. And what we found was that all the transition states, irrespective of the starting point, were lined up on a particular value of the z vertical translation. So this indicates that translation in up and down of the helical bundle is a major coordinate. By contrast, we saw considerable scatter even within one path sampling program in the direction of the angle. So the angle appears to be less important to this coordinate. And but most importantly, the two paths used to see transition path sampling gave very consistent result. What you also see in these transition state structures, as I've already pointed out, is hydration. And if you look carefully, you can see that we see a diversity in the hydration of the transition states. And this, in some sense, of course, also connects to what we saw before in Mark's beautiful presentation. Hydration is a very variable and strongly dependent on local geometry polarity. And so what we saw here is a fraction of transition state structures were occluded. That is, you had the ion buried, maybe with a few water molecules tagged along as it was passing through the membrane domain. And in others, we actually saw continuous water chains, albeit at the single file level, connecting the two membrane sites. Now, you may worry that these states could result in leakage, but you should keep in mind that the formation of these conducting states are at the rate of, let's say, 1 per minute or so, and they are also in the MD simulation, they're highly transient on the order of 1 nanosecond, so there's absolutely no proton leak issue here, even if you transiently have a conducting state. So let's look at the transition states and their dependence on the ion to be transported and on temperature. So what we did is we swapped out sodium against protons. So we took aspartate carrying the sodium and we protonated it, removing the sodium, and vice versa. And we changed the temperature. So take a structure harvested at 37 centigrade, transition path sampling, and then constrain it, relax it at 100 degrees centigrade, and then use it as a starting point. And what we found was that the transition states where transition state characteristic was maintained both by exchanging the ion and by increasing the temperature. Transition states for protons are also transition states for sodium, and transition states at 37 degree are also transition states at 100 degree. So here now you see a movie that puts all the simulations together. And just to emphasize once more, these are unbiased molecular dynamics simulations. So there's no force pulling the transporter domain. It's just, as I said, a judicious choice of initial condition where this happens spontaneously, where the transporter domain moves up, tilts a bit, and exposes, in this case, in yellow, the sodium ion to the outside, and we can actually also see a spontaneous release of the ion. For the reverse path, we have to use a bit of alchemical trickery because in these calculations, we did not use QMM type of modeling. Instead, we just protonate the aspartate, and then we can actually see the reverse transition path that now takes the blue transporter domain, again, tilts it moves it down by a few angstrom, and then exposes the axis, changes the axis state from outward to inward open, as has just happened, and now the proton can be released. So here is a cartoon version now of this. So we have sliding domain that through an aspartate carries the ion and the proton up and down. That we could simulate, but we also noticed something interesting that goes beyond the default mechanism, and that was that above and below the ion binding site, there are residues which are quite hydrophobic. In particular, above, we have a tyrosine and an isoleucine, and reliably in all transitions, they form a gate in one axis state, being locked against each other, and this gate opens in the transition to the other axis state. And so this was quite interesting. And when we then used this as an input in an effort to construct a reaction coordinate of this process, what we found was that it could predict the observed committer values to a significant degree. And actually, in an optimized committer representation that used as possible inputs vertical displacement, angular tilt, and gate formation, we found that we did not need to include the angle of the tilt, which is consistent with the picture I had shown you before, but gate formation was as important as an indicator of the state with respect to the commitment as was the vertical displacement. And this is the ability to predict committers on structures that we had not been seen in the training is illustrated on the right. I would say while the prediction is not 100%, just being able to in this sea of confirmations to find structures for a transporter with a 1 per minute or so transport rate that have a 50% chance of going either way is already quite a remarkable feat, I think. So now back to these hydrophobic gates, and they're not just important for the mechanism of the transport, but they have direct impact on the transport rate. And this was shown by our experimental collaborators in the Kühlbrand lab, by David Wöhlert and Özkan Yildiz. And what they found is that mutations of the gate residues to alanine retains the function, so this is a perfectly functioning transporter, but actually the transport speeds up. So the gates apparently lock not just the axis side, but they slow down the transition between the alternating axis states. By removing them, you make the transporter faster, so we have a gain of function. However, we suspect we could not show this. It results in a loss of fidelity of the transport. So in summary, we have a, I'm sorry, we have, I think I just lost my presentation. We have a dominant elevator motion with a bit of rocking and with hydrophobic gates sealing the ion binding site as the mechanism. Now, one of the interesting questions, also of course the ion channel community, is selectivity. And here we have a family of transporters with a high selectivity for sodium. Now we look at the closely related methanococcus channel transport MJNHAP1, where selectivity has been demonstrated experimentally, and it does not transport potassium, it is transporting only sodium. So we looked at this again with molecular dynamic simulations. We found that we had again spontaneous ion binding to the residues implicated by the structural studies on the PA and HAP transporter, and homologous residues. And we looked at this both with regular classical thermodynamic simulations and with a QM-MM type framework, finding consistent ion binding structures, both for sodium and for potassium. And as you would expect, the hydration state is a bit different when you go from sodium to potassium, but there's no, the coordinating residues are not substantially different. So this raised the question where the selectivity for sodium is coming from. So to address this, we performed free energy calculations by interconverting sodium to potassium in an inward open state, an outward open state, and in bulk solution. And what we found was that in the inward open conformation, we had a very high selectivity for sodium, calculated delta G of about 5 kilocalories per mole. In the outward open state, selectivity was significantly lower, about half a kilocalorie per mole. And so this is going in obviously right direction, which is very encouraging. We have sodium selectivity, but there is this asymmetry, which is a bit unexpected. But we think, again, without having any direct evidence, that this makes a lot of functional sense. The carcass is in a saline environment where the cell interior is high in potassium and low in sodium, and the exterior is high in sodium and low in potassium. So it would seem to make a lot of sense to be selective for sodium on the inside to prevent the loss of any potassium ions. Whereas on the outside, selectivity for sodium is not needed because sodium is, of course, highly abundant anyway, and any chance uptake of potassium would probably be even welcome by the organism. So the real proof of an understanding would come if we could use our calculations to engineer a transporter that were selective for potassium instead of sodium. And we tried that. We made a series of mutations. These are alchemical mutations. And we could, by double mutations, go to revert the selectivity partly, go from 5 to 2.5 kilocalories per mole. But as was also confirmed by experiments, that was not sufficient. There was no detected potassium transport even in the double mutant. So this effort unfortunately failed. So we don't know what exactly happens in between the access states. So there could be other selectivity mechanisms, not just the ones associated with the binding. So the last few minutes, I will just skip over some work on active transport powered by ATP, where we look at substrate translocation in an ABC transporter. And here we build on absolutely beautiful work done here at the Institute, when Arne Müller and his group were still here. Is now having his own group in a collaboration with Arne Müller and the Robert Campe group at Frankfurt University. So they have been studying PMR-AB and ABC transporter, again, of a hyperthermophilic organism. And what we could do with the ND simulations was take this gallery of intermediate structures and look at them in some detail. And I just want to highlight a few things that where simulations could contribute. One of them was to look at what powers the conformational change. And here what we found was that both experimentally and computationally, that phosphate release very likely is an important player driving the conformational change. And in this picture, you can see the presumed exit path or is one of the two exit paths for phosphate release. And we could also establish a connectivity between the conformations, as seen in the cryo-NM structures, which are collected in part under turnover conditions. So we were looking at ABC transporters in active turnover, and then complemented by DMD, develop a relatively complete now picture of the transport and catalytic cycle. And I don't have the time to go through this in detail, so I just want to highlight this last point that I already brought up, the importance of phosphate release, a PI release, as a driver of the conformational dynamics. And here it is driving the restoration of the inward-facing structure ready to take up substrate for the export of this ABC export system. So with that, I would like to conclude. I started out with a warm-up PF mate simulation showing lipid flip. I then gave you a fairly detailed view of sodium proton exchange in PNHAP, and showing the power, and hopefully convincing you of the power of transition path sampling, also for these very complex transitions. The mechanistic insight that we gained, or at least in part validated by experiment, so this is the importance of these hydrophobic gates. I looked at ion selectivity and showed that at least we can capture part of this, and showed that NV simulations also help us gain understanding of ABC exporter system. That brings me to the acknowledgments. I would like to thank in particular Reza Medipur for his work on PF-MATE and also on TMR-AB. And I already mentioned the collaboration with Shahar Safarian and Hartmut Michel. The PA-NHAP work was a collaboration on the experimental side with the labs of Werner Kuhlbrand, with a lot of beautiful work by David Böhler and Özkan Yildiz. And on the computational side with Keiichi Okazaki, who is now in Okazaki, Japan. And much of the work was also done by Judith Warnau. She graduated last year, was co-supervised by Ville Keiler, who also played a very important role in the ion selectivity study. And I already mentioned our experimental collaborators for the MRAB. Last thing I would like to point out, we have openings for postdoc and PhD positions. If you're interested, let me know. With that, I would like to end my presentation and take your questions. Thank you.
Thank you very much, Gerhard. It's a very beautiful talk. There are several questions from the audience. So Momar Bukamsha asks, is there an important role concerning the Fick laws for the diffusion and dynamics of living molecules to simulate this type of scientific research?
Yes, so diffusion is, of course, critical as an approximate but remarkably accurate description of both the transport of substrates, but also of the conformational dynamics. So the power of diffusive models, I think, is enormous. They require refinement, but what we found was that, and this is not included in my presentation, that even the conformational dynamics of the exchange event itself can be described quite well by diffusive models.
Thank you. So Lucille Lemotte is asking, you mentioned transition path sampling as a way to get unbiased sampling, but the way the seeding is done is based on a reaction coordinate choice. So this needs to be done carefully in a system-dependent way. Can you give details on how that was done in the case of the proton-sodium exchanger and comment on what is important to consider for other systems?
Yeah, so Lucy, you're absolutely right. that the time evolution trajectories are unbiased, but somehow we need to come up with starting points. And of course, this applies to every molecular dynamics simulation. The starting point is always biased. It's something that you constructed based on input. And here, of course, it is particularly important to be careful about the choice of starting point. And I don't think there are general recipes in transitions between clear reactant and product states with reasonably well understood delineations. What one can do is use trial coordinates that drive the system forward and trial coordinates that drive the system backward. So, and that's what we did. And in that way, we have starting structure. These paths tend to be somewhat different because of the poor choice of coordinate and the limited time of them. So you have different hystereses, and then run multiple transition path samplings in parallel and see if the characteristics of the paths are actually converging. And I think that's some evidence that the bias of the initial choice has gone away.
Okay, so Igor Bodrenko is asking, a forced selection of initial condition is also a sort of biasing, isn't it? Why then calling this method unbiased simulation?
As I said, every simulation has to choose an initial condition. And in that sense, you're always biased by the choice of your initial condition. And the important thing is that when you actively pull, you have essentially a guaranteed outcome that if you pull hard enough, you'll get wherever you want. If you let something evolve freely, you have a chance of discovering something new. Transition path sampling is, in that sense, it's a discovery tool of the mechanism.
Okay, so Mark Sanson is asking, does the gate in mutant I163A, Y255A become more leaky?
Yeah, this is, again, an excellent question. I don't have the numbers on top of my head, but I believe there is, in the simulations, there is a slight change in the hydration character. And experimentally, unfortunately, this was not possible to make a very extensive characterization to see if there are longer-lived leaky states. In the experiments that were done, there is no evidence for any kind of leakiness. But I mean, of course, the suspicion is that this is the likely reason why the organism sacrifices a bit of speed, factor 3, to prevent some other detrimental effect on leakiness, is the likeliest of these, but we have no evidence for it, except that in the MD simulations we can see changes in the hydration.
Okay, so there are other questions by Roland Roth and Ben Corey, but I think you can answer in the private chat. So because we are already running late, unfortunately. So we thank Gerhard again.